So I'm Julia Rozenfeld, Associate Director at Teva Pharmaceuticals, Biologic Discovery, Sydney, Australia. And my talk today will be focusing on early immunogenicity prediction and why actually it became so important component in drug discovery. Let me introduce you to Teva. Teva has been around for more than 120 years, and it started as a small pharmacy. So the company doesn't survive for so long just by chance. It survives because it survived because it's constantly improved, was constantly evolving. And what motivates us today actually motivated people 120 years ago. It's to deliver good medicines to help people's life. As you probably know, Teva is well known for its leadership in generics. But over the years, it was transforming with a strong emphasis on innovative medicines. And it's well reflected in our current pipeline. As you can see, you may have heard about recent partnership with Sanofi about duvakitug, anti-TL1A antibody, best-in-class antibody against ulcerative colitis and Crohn's disease, which followed by another collaboration with antibody against anti-IL-15, against some autoimmune diseases. So these both antibodies were discovered and developed in Sydney, Australia. They are both best-in-class antibodies. So it's very exciting to work on best-in-class antibodies, but it's also challenging because we should predict and expect from early on what challenges can expect us during the development of the antibody. And as you all probably know, the most expensive surprise in drug development are immune surprises. So we can develop an antibody that will meet the target biology, that will be extremely potent, but then it comes to the clinic and it has a challenge with ADA production. Keep forgetting what to press. And it's not a rare situation, as you all know. Here you can see the very efficient drugs, but they all have one thing in common. They all cause immunogenicity by elevation of anti-drug antibody production, and which led to loss of drug effect, reduced therapeutic activity, and even discontinuation. And that's the reason why assessment of immunogenicity is a very important component of drug evaluation. So when do we need actually to check the immunogenicity, or why do immunogenicity first comes in the place? We would expect one factor. You've probably seen this slide already as a differently illustrated in many other presentations. But immunogenicity is a multifactorial issue. It can be caused by drug-related factors, such as related to the sequence of the drug, and be caused by post-translational modification or presence of T-cell epitopes. It can be related to the patient, especially to MHC haplotype, ethnicity, age, and etc. It can depend on the disease state and be related to administration route or autoimmune disease state, and it also can be related to the process-related factors such as storage conditions or contaminations from host materials. All of these factors emerge at different times, and that's the reason why no signal assessment is enough. So when is better to assess immunogenicity? And the question is, if we would like to measure immunogenicity, we need to do it in the clinical stage. If we would like to avoid immunogenicity, we need to assess it at preclinical, because at preclinical stage, we still can manipulate the sequence and change the sequence. And most successful strategies combine actually both. They will assess preclinical risk. They will assess it at preclinical risk and mitigate the risk. And also they will check or monitor anti-drug antibodies during the clinical trials. So I'm actually an antibody engineer, and my butter and bread is optimization of antibodies. So I will focus on preclinical assessment. Okay, it's another slide that you've all seen, a bit differently illustrated. So the therapeutic antibodies can be uptaken by antigen-presenting cells. It will be it may be processed in the antigen-presenting cells. It may be presented by MHC class 2. It may be it may activate T cells, and it may cause anti-drug antibodies. The companies such as ProImmune can actually validate any of these steps of anti-drug antibody production. At Teva, okay. Yes, thank you. At Teva, we manage immunogenicity by design. So we screen the antibody sequences for immunogenicity risk. We identify potential T cell epitopes, and then we remove these potential T cell epitopes by molecular engineering. Up to date, we screened over 1,600 sequences. And we could find more than 1,000 sequences with potential T cell epitopes. To manage the immunogenicity risk by design, it's essential to make sure that the antibody will reach the market without any competitive disadvantage. So let me give you an example of the recent program that we've done. We screened 197 sequences across 8 months. So we found 104 potential immunogenic sequences. Through antibody engineering, by looking at binding, function, biophysical characterization, we have done immunogenicity assessment of all of the different variants to remove these potential immunogenic epitopes. And by removal of these potential immunogenic epitopes, our lead antibody had 0 T cell epitopes. So you would ask me, is it worth the time? Is it 8 months? Is it worth all of the money that we actually put into the project? And I would say yes, because it's, as you can see, this was well reflected in our clinical programs. All our antibodies in the clinical programs got very good half-life times, higher than competitors with very low antibodies production, 2 to 5%. So it means consistent, it's supported evidence of low immunogenicity. As I mentioned, the most expensive surprises in drug development are immune surprises. And because we invest early in the drug development, we invest early to give our molecule the best chance. By making this molecule to the market, we are coming to we are making it to the patient. And by this, we are making a difference in people's life. And thank you for your attention.
Thanks for the nice presentation. Do you do the in silico analysis in-house, or you take the help of the vendors?
In silico analysis is done at home, in-house.
Okay. So this, whatever the in-house generated data, has it been submitted to the agency and it's been accepted for any of your programs?
So mostly it's also supported by in vitro. After we do in silico, we are doing some in vitro.
Mm-hmm. But still, you will have to publish quite a lot of in silico, the background data, the validation of the software which you are using. Has it all been complied with the agency's expectation?
But again, it's in silico is supported by in vitro. So in silico work, it just guides us, and then we confirm with in vitro studies.
Okay, thank you.
Hi, Julia. Thanks for a very nice talk. In making drug, we say the quality by design. It's really design making the final quality of the drug and the effects. So I'm wondering, like, for example, you already know like the sequences that could trigger T cell response or immunogenicity. But what if like the sequence is really important for the drug effect, but still it causes immunogenicity? What should we do?
So when we discover the antibody, we already have the sequence, right? So we know the sequence of the antibody. Then we do analysis of the sequence, and we start applying all of different modifications to the sequence, because the main concern is to look at the sequence liabilities, to look at the T cell potential epitopes. If we need to do affinity maturation to make the antibody more potent, and etc. So all of this comes into the circle of it's a repeatable process. I forgot to mention about this. It's a repeatable process. So we need we have a sequence, and then we kind of we are doing modifications to improve the sequence.
Thank you.
Is any questions? No?
Thanks, Julia. And we talked a little bit about kind of the stringency for in silico, and I guess based on other risk factors, would there be a time when you might look at more sequences and try and be more stringent, or indeed with lower risk, less stringent in how you view the epitopes?
Yeah, so look, frankly, in silico, more educates us about the immunization processes. So we will not exclude the sequences initially just because they were flagged in silico design. Because again, we are after the potent antibody, so we don't want to exclude all T cell potential epitopes and get nothing from the drug. We still need to have a potent drug. So it's kind of we try not to exclude anything from the start, but with with the time, yes.
Yeah, that makes sense. Thank you.